Hello, I'm Nathan Hendricks. I'm a specialist at MAST. I'm here to talk about my poster, which is looking at the proteomics of SIBO. So I'm looking at SIBO, which is small intestinal bacterial overgrowth. It's a GI disorder which occurs when you have certain select species that are overabundant. People have looked at the sequencing, genomics of SIBO, but nobody's really explored the proteomics of SIBO. So we're going to look at the protein makeup of people with or without SIBO and see what sort of diagnostic or prognostic markers there are there. So we're looking at small bowel lumen aspirates from the Reimagine study. So we took 20 microliters of these aspirates, and it's important to note that this is the centrifuge aspirate. It's not the tissue. And we took 20 microliters of that and we processed it by trypsin digestion to prepare it for proteomics analysis. We took these processed samples and we analyzed them with data-independent acquisition on the Xenotoph 7600. So of the proteins we see many IgGs, which is something we might expect to see in SIBO, and we also see defensin A1 and defensin A3 are one of the most significantly upregulated proteins in SIBO, and this all matches with what you would expect to see in an immune response to infection. So if we look at the particular pathways that are in this data set, we have many proteins that are part of the neutrophil degranulation pathway, and this highlights that there is a specific neutrophil response occurring to SIBO in the lumen. So to summarize some of the important results we see immunoglobulins and defensins in our protein data set, which is something that we'd expect to see in response to bacterial overgrowth. We also see a lot of activation of the neutrophil degranulation pathway and proteins related to neutrophils, and this all suggests that we are seeing neutrophil activation in response to SIBO. So thank you for listening to this summary and thank you for checking out my poster. I hope you check out all the other posters and presentations that we have from MAST at the 2026 DDW.